As I was saying, some of you will have heard this talk before. Many of you will have heard pieces of it if you go to the M&M talks that we have, the kind of combined group meeting that we do. There we go. Just gonna, yeah. So if you have seen this before, my apologies, but I know many of you haven't. So this is work that we've been doing in my lab, trying to understand empirically this picture. That's a classic picture, but we'll get to that in a second. So yeah, before I move on, I want to thank the people who actually did most of the work I'm going to be telling you about today. Brianne Sparta was a postdoc in my lab for many years and did a lot of the stuff I'm going to be mentioning. And then Tim Hamilton, who's here, started off in my group as an undergraduate and now is in the PhD program in bioinformatics, one of the components of this series. So that's really exciting. Okay. So everybody knows about this technology that's been revolutionizing how we think about cell state and transcriptional data regarding cells. But I want to highlight something about this technology that I think is sometimes not often appreciated, or at least not we're not reminded of it. So historically, if you wanted to know what was going on inside a complex tissue in terms of the transcriptional response of the cells, or their molecular state, you were forced to use oftentimes slices. So you would go in, you might freeze or fix the sample, slice it very thinly, and then use a microscope along with, say, something like fluorescent in situ hybridization or protein antibody staining, things like that, to try to figure out what's going on inside those tissues. Say you have a diseased animal, you try to figure out, okay, what is the cells that are underlying this disease? What are they actually expressing? And in this sort of process, you were able to kind of retain the morphological and structural characteristics of cells when you were actually doing those kinds of experiments. So it was very powerful. But the problem is that you can't actually tell very much about what's going on. You can only look at a very few number of genes at a time. So this technology, single cell sequencing, has revolutionized our ability to sort of track those transcriptional changes because we can look not only at all the cells in very complex scenarios, not just tissues, not just slices, but whole organisms in many cases. We can get data from millions of cells, but we can also look at the entire genome. And recently, spatial transcriptomic technologies developed by Roy and others have brought us back to being able to sort of track those structural characteristics. But the vast majority of data that we have still comes from these kind of experiments where you take a tissue, you dissolve it, you put it through some sort of procedure. There are several. This is an old slide, but there are many different ways of doing this now. And essentially what this allows you to do is characterize in every single cell in this complex tissue, what are the expression levels of all of the different RNAs in the genome. And so that's a really powerful technique. But what I'm highlighting here is that when you do this process, you lose all of those traditional morphological characters or other structural indicators that were used traditionally to indicate cell type or the composition of the tissue. And so after you've done this experiment, you can tell, the expression level of different RNAs in these different cells. But when you want to recover cell identity, you do so computationally. So we had a great talk last week about that. You probably all are very aware of this. Typically, one employs a kind of clustering strategy to go in here and actually determine what the cell types are in the data set that you're looking at. And you'll often see, and many of you will have seen this either in my talks, if you've seen any of them, or in any talk about single cell RNA sequencing, you'll see these beautiful pictures that people generate. And they're really, really compelling. What this is data on this freshwater cnidarian called Hydra. It's a fantastically interesting organism. It's immortal. So as far as we know, it doesn't die of its own. It's like an elf in Tolkien. Not only that, I mean, it's not as pretty, right, as Legolas, but it's pretty cool. And they can also, if you put them in a blender and chop them up and dissociate them, they will spontaneously reassemble, like Dr. Manhattan. It's nuts. So it's really, really cool organism. And, this is just one example. People have done this for all kinds of organisms. But you take this thing, you dissociate it. In this case, they had about 20,000 cells they were able to characterize using single cell RNA sequencing. And then they clustered those data and displayed it in this two-dimensional representation of the experiment here. This is a t-SNE plot. We'll talk about that later. But it's a really interesting kind of thing that you see here. And it's a very common way of kind of looking at this data. What you're looking at, every dot is a cell. and they're colored according to the clusters that the researchers in this case identified using a computational pipeline that's very standard in the field these days. And one of the things about this picture that's very, very satisfying is that all the colors are close to each other, right? So you see here that all of these progenitor stem cells are near each other. All of these neurons colored blue, this fetching light blue here, are all next to each other. And so that's very satisfying. And the question is why? Why do we expect that? Why should we think that this is what we're going to get? And that comes from this really long-standing problem in biology, the biology of metazoa and all multicellular organisms, which is how do you develop the huge diversity of cell types that we have in our bodies from a single cell, a zygote. So the cool thing is that we all start out as a single cell, no matter what kind of metazoan you are, a human, mouse, hydra, whatever. You start out as a fertilized zygote, but then through the process of development, you give rise to things like muscle cells or neurons or these neutrophils here, and or monocytes. And the idea here is that these cells are all very, very different phenotypically, right? No one would ever confuse a muscle cell with a neuron or a monocyte with a neuron, right? These are very, very different. And the kind of paradox here is that in genetics class, right, we're taught that there's a correspondence between genotype and phenotype. And originally, August Weismann, working in the late 1800s, tried to explain how it's possible to go from one thing and produce all of this diversity by saying, well, actually the information that is present in the zygote is asymmetrically distributed throughout all of these lineages to give rise to these different fates. He called it the germ plasm, and that germ plasm was given to the lineage that gave rise to the gametes early on, but then after that, it was sort of incontrovertibly converted into these different types of structures. And later on, we found out that while that's a beautiful theory, it's completely wrong because the DNA that's present in every single one of these cells is the same. These are all clones. And so it can't be that they have different sort of genetic information. And so this problem was actually solved. It wasn't directly solved. It's a very complicated history, but the modern explanation that we have for this is due to Conrad Hall Waddington, who was working about 50, 80 years later than Weismann. And his idea here was actually that there's an epigenetic landscape, an epiphenomenon to the genome that gives rise to all of these structures. This is the first picture that he drew, but this is the more common one. And the idea here is that you start off with a zygote, and then during development's coming into the plane. How many of you have seen this picture before? Many of you will have seen it, so you're aware of it, but I'll just go over it. The idea is you start off, say, as a zygote, and then during the course of development, all of these slices correspond to different developmental time points. You reach these bifurcation points where the zygote undergoes a differentiation. It sort of transitions into two different lineages, and then this lineage goes down here and maybe eventually will split and form like muscle cells and stuff like that, whereas this will go and form neurons. And so that was a very powerful picture, and it's evolved into a very sort of beautiful, but I think often not super well appreciated theory of how cell types are specified. And it's a theory that actually has been in conversation with dynamical systems theory in math for a really, really long time. In fact, if any of you have ever taken a dynamical systems theory class, you will have heard this concept of a basin of attraction, right? So where does that word come from? It actually comes from this picture. So the idea of basins here in that theory was actually sort of developed in part through conversation with Waddington's work. But anyway, the basic idea here is that in our genome, we have all kinds of genes, and many of those genes either directly or indirectly influence the transcription and production of other genes. So this forms a gene regulatory network. And if we make or conceptualize that gene regulatory network as a dynamical system, the state variables of that system are going to be things like the concentration of the RNA and protein. And ultimately, what's happening here is you start out with a zygote, and then during development, there are bifurcations. So these are fundamental changes. Some parameters in the system are changing, and you develop these different attractor states, which are equilibrium points in dynamical systems, and those attractors correspond to the different cell types. So the basic idea here is that the sort of specification of these very distinct cell phenotypes comes from a process whereby gene regulatory networks are evolving during the course of development, not evolving in a sort of EEB sense, but just evolving in time. And then at the end of development, you have these well sort of defined valleys down here, these attractors and that correspond to different cell types. And the idea here, the fundamental idea is that the concentrations of the various RNAs and proteins should be very different between these different states. This gene, the gene regulatory network that underlies this landscape generates an attractor over here, and that attractor is distinct from this one. So there are different genes being expressed in different amounts between these different states. And that's a beautiful way of explaining how this is possible, because for one, it explains sort of mechanistically how you're able to generate so many different states from a single starting point. But also the fact that these cells express different amounts of different proteins, that makes sense, right? That allows the cells to respond differently to their environment, to have different structures, to have different phenotypes. So it's a beautiful and natural idea. And so one of the things that this theory predicts is that the cells should cluster in these different cell states that are characterized by an epigenetic landscape, which is fundamentally transcriptional. So this is why when we look at the kinds of pictures that we see and we see these different clusters, we get very excited, right? Because the idea here is that, neutrophils might be down here, monocytes over here, neurons over here, whatever. And so they should be sort of different in the gene expression space corresponding to these different attractors. And so, yeah, so this looks exactly like what we would expect. So that's really, really good. But there's a bit of a problem here. And that problem is that if you look at this carefully, and sometimes when I show this to students who really aren't familiar with this, they'll be like, well, what is this Tisney thing? What is this axis? What does it represent? And that's a really, really interesting question, because the attractors that the theory predicts should exist in the gene expression space that we're talking about here. So in a single cell RNA sequencing experiment, you start out with all of your cells, you go through this whole experimental rigmarole and all of this alignment steps and stuff, and then you generate a UMI count matrix. And this matrix, we might represent it with every row as a cell, every column as a gene, and the entries of this matrix is the number of unique molecular identifiers for that gene that were found in that cell. So you can think of this as the number of unique RNAs that were captured for this gene in that cell. And if we think about this, this sort of matrix represents every cell as a point in a really, really big vector space. So typically in these experiments, you'll have anything from 20,000 to 60,000 genes that you'll have information for. And we can talk about why it's more than 20,000, but whatever. The point is that this is a really high dimensional form of data. And you have to do a lot of stuff to this data to take it from 20,000 dimensions and produce a two-dimensional representation, right? So you start with 20,000 genes, and then there's all of this dimensionality reduction that happens. We'll talk a little bit about those steps in a bit, and that's what gives you this actual picture, right? And it's universally the case, it's not just common, it's universally the case that dimensionality of this data is significantly reduced before you do any of that analysis to produce these computational models of cell types. And the question that we first started out with in my lab was, can this cause any problems? So I started working on this when I was still at Kansas. This was in the early days. I had a friend named Peter Sims who was involved in developing this technology, sent me some papers. I was like, this is really cool. But I was reading the computational methods and I was like, whoa, there's a lot going on here. This is a complicated process. So how, what is going on here? And can all of this dimensionality reduction cause any problems? And the example that I give for why this could cause problems is this very familiar situation we have of taking the three-dimensional globe, the Earth, and representing it as a two-dimensional map. So all of you will have seen this picture at some point in your lives, right? Because a globe is really nice, but the problem is you can't hang a globe on the wall of your classroom, right? So if you want a two-dimensional map, you've got to do something. And in doing this, there are two serious problems that you face. The first one is that area is distorted. And this is kind of a common criticism, especially a few years ago. People got really excited about talking about this, of this particular version of that map, which is called the Mercator projection. And what you can see here in the dark colors are all of the countries in their actual sizes. And in light blue is the way they appear on the map. And you can see here that for countries towards the north of the globe, especially Greenland, there's this massive distortion of area. So, Greenland in the picture here looks larger than Africa, but it's actually only about 1/10 of the size. So that's one problem. But there's actually a bigger problem, and that is that you have to cut somewhere. And this is a theorem. You have no choice. You've got to make some kind of cut somewhere. And I don't know, I'm old, but this in the when Sarah Palin was running for vice president, there was this fantastic, and I actually can't tell the difference between Sarah Palin and Tina Fey anymore. When I think of Sarah Palin in my head, I actually think of Tina Fey. But whatever. The point is that in this skit, Tina said, "I can see Russia from my house," and we all thought that was really funny. It was a statement that Sarah Palin made, and people thought, "Oh, you know how intellectually challenged is Sarah Palin?" But actually, the reason we thought this was so funny, I would say, is that everybody sits around and thinks that Russia and America are on opposite sides of the world, but they're not. Russia is actually one of the closest countries to America. Alaska and Siberia are really close to one another. They don't have a land connection, but part of America is closer to Russia than it is to anything in South America, right? And so this is something that I think highlights how these representations that we use when they make things like cutting through the map here and putting things on different sides of the world, we carry that around in our head, right? And that can influence how we think. So there's a problem with dimensionality reduction. So tools like Disney and UMAP do a pretty good job for simplifying two-dimensional surfaces. This is the common thing you'll find if you go to the scikit-learn website and learn about these dimensionality reduction tools. They'll show you how they do on this S-curve, which is literally just a piece of paper that's been curled in 3D, and it can flatten it. It does okay with that, but it really is terrible at any other problem. And I can go on and on about this. We've done a lot of work in my lab characterizing how these nonlinear dimensionality reductions work, these tools work. But you can see here that what's happening here is that you've colored all of these points by where they are on the S-curve, sort of from one side to the other. And you can see that that rainbow pattern is largely preserved. But here we've done the same thing with a sphere, and you can see that that rainbow pattern is severely disrupted. And in some sense, it's impossible for Disney and UMAP or other tools to represent this sphere in 2D without doing that, because it's just mathematically impossible to maintain that pattern. So what we did originally was develop kind of a metric for quantifying how much this sort of cutting is influencing these pictures that we look at. So here's a simple example of a sphere that we're reducing to 2 dimensions using Disney. On the far left side here, we have a single point, and we've colored all of its sort of 100 nearest neighbors red, right? So those are the original near neighbors of the point. You can think of that as being like Alaska and Russia. And then for that same point, in the 2-dimensional representation, we're going to color all of the points blue that are the 100 nearest neighbors of that point. And then we can look at the overlap between that. And it turns out that if you do that, you can actually characterize how dissimilar these sets of neighbors are using a simple quantity called the Jacquard distance. So this just quantifies how different those two sets are. So when this number is 0, that means that the sets completely overlap. There's no distortion. But when it's 1, that means these sets are disjoint. They're completely different. And we can do that for every point in the data set. And we'll talk about how we choose the number of nearest neighbors in a minute. But you can do that for every point in the data set. And then what we do is we just average this quantity across the whole data set and come up with a statistic we call the average Jacquard distance. And so what we can do here is then apply this idea to these kinds of pictures and sort of see how faithfully do they represent the nearest neighbor relationships that are present in the data. And one of the reasons we were so interested in nearest neighbor relationships is that if you look at clustering algorithms and other analytical techniques that people use to actually produce things like cell type clusters and stuff like that, they're all based on nearest neighbors. They all use kind of a nearest neighbor sort of calculation as the first step in the analysis. So it's really interesting to say, okay, well, how well does this actually preserve those nearest neighbor relationships? And what we did here, and this is just one piece of work that we've done to kind of look at how these standard pipelines end up affecting the data. You're comparing the raw data. This is the actual count matrix you get from the experiment. No one ever looks at it, but it's actually your data. And then people will typically choose a subset of, say, 2,000 highly variable genes that they think are important. We'll talk about that later. But basically, this is a subset of genes. They then normalize the data and perform a log transformation. And then they do PCA. This step is where people typically cluster. And then this is sort of a UMAP or a TISNE. This is the step where people visualize the data. And one thing that I want to highlight here is if you look at the entries in this heat map here, they're often about 0.95, very close to 1. So for instance, if we compare the raw data to the PCA space where people do their analysis, that's 95% different. And that's a pretty big distortion. So that means the vast majority of neighbors are not preserved between those two spaces. And even if you look at PCA compared to UMAP, you've got 80% distortion. So a lot of the nearest neighbor relationships that are present when you analyze the data actually are no longer nearest neighbors when you actually look at it. And so one thing that you might ask is, well, okay, this is fine, but in this particular heat map, we're choosing 20 neighbors. It's a very local notion of neighborhoods. It may be that these numbers are really high and all of this distortion is there just because you've kind of rearranged the kind of local relationships. You've sort of permuted. We don't really care if you don't get the specific relationship between, say, San Diego and LA correct. As long as you kind of have a picture of SoCal, you're good. What about sort of more global distortion? So what we've done here is just to represent the sort of locality of this distortion is change the size of these nearest neighbors that we look at and then compare the raw data to a variety of different, like the TISNI, the UMAP, or PCA. We've done a very extensive sort of analysis of this. I won't go into it, but the point is that you see massive amounts of distortion even when the neighborhood sizes are like 10,000. So what this means is that points that are very close to one another in the original data are on completely different sides of the map, much like Russia and America when we do the Mercator projections. So that's really interesting, right? And one of the corollaries of that finding, and this is a finding that we made a long time ago, and then Lior Pachter's group did a really great job of sort of explaining how this might influence how you interpret these visualizations and what their utility might be. There's been some interest in this in the years since. But basically, one of the things that we noticed is that what this finding means is that we got really excited because all of these neurons here are all in the same part of this or in the same place in this picture. But if you've got nearest neighbors here that are neurons, or even fairly nearest neighbors that are neurons, fairly close points, it means that they're actually in completely different transcriptional states. And that's kind of interesting because the whole reason we got excited about this was that things that were in the same sort of cell type were in the similar regions of this representation of gene expression space. So we actually decided that it would be really interesting to go into the raw data and look at its structure, to actually try to find if we could see these attractors. And this is, I don't know if any of you went to the picture or to the talk last week, but there was a picture that was essentially identical to this in that talk. So people often have in their minds when they're clustering these data that it would look like this. But those of you who have actually done, how many of you have actually tried to cluster single cell data? A few of you. It's not necessarily easy, right? We've tried to do it in my lab. It can be very challenging, and we could talk about that if you're interested. Why that? Well, you'll see why that is perhaps in a minute. But basically, the point is that this is the picture we carry around in our heads when we do clustering. And so the question is, can we actually test whether this structure is present in the data? And to do that, Brianne and Tim and I developed a very simple approach. Those of you who might know any computer science, in computer science, this is called random geometric graphs. We call it epsilon networks. But the basic idea is that for every cell in your data set, no matter the dimensionality of the space, you can take each cell and draw a little ball around it of radius epsilon. And we're going to change that radius, as you'll see in a minute. So we draw this little ball, and what we do is we say, okay, there's this radius epsilon. We draw it around all of the cells, and then any two cells that are closer to one another than that distance cutoff, we put an edge between them. We kind of connect them. They're sort of neighbors at this particular epsilon. And so when we do that, when you have a small epsilon, you'll see that only very near cells that are nearby in the gene expression space get connected. And what we can then do is track the sizes of the components in graph theory. So a component here is just a group of cells that are all connected to each other. So this is a component, this is a component, this is a component. And you can look at all of the components, but what I'm going to show today is just tracking the size of the biggest one, because it's a really easy thing to do. And that biggest group of cells that are all connected together is called the giant component in graph theory. So we're gonna be tracking the size of that giant component as we change this radius epsilon. And if this picture that we often see schematized for RNA sequencing experiments is true, what we should see is that as we increase this epsilon, we should connect these groups of cells together that correspond to these sort of regions of gene expression space where sort of cell type A or B or C lives. So we should put them all together. And then one component of all of these pictures that people draw and a prediction of Waddington's landscape is that there should be white space in between these groups. That corresponds to the hills on the landscape. So when we encounter that white space, what we should see is that as we increase epsilon, we put all of these cells in these local groups together. If we track the biggest one, we should see that at some point it stops growing, because as we increase epsilon, we've reached one of these white space regions. We're no longer going to be putting anything together. But then when suddenly we increase epsilon beyond the sort of size or the sort of width of that white space, what we should see is that all of these cells here in purple join this blue group all at once, right? So what that should correspond to is a discrete jump in the size of the giant component. So even if we don't know the identities of the cells, as long as they're organized in this sort of structure that I've shown here, we should see these discrete jumps. And those of you who have ever heard about or used DB-scan or HCB-scan, those are clustering tools that rely on this idea. So this is not a new idea, it's just something we used to characterize the structure of the data. So you should see that you get these three steps corresponding to these three cell types, and you could do things like characterize the radius of the cell types, the number of cells, et cetera. So this is a beautiful idea. And we applied this first to a very old now data set, but one that's very, very nice, because what the people, this is actually from the 10x company themselves, one of the main providers of single-cell RNA sequencing kits and instruments. What they did in this particular experiment is they took peripheral blood monocytes from humans, these are just white blood cells, and then used a traditional process called fluorescence-activated cell sorting, where you basically label certain cell surface markers on these cells using fluorescent antibodies, and then you can use the fact that B cells have certain markers that they express, T cells, et cetera, to sort of sort them into different tubes, and then they sequence those tubes independently. So we have single cell data for pure B cells, pure NK cells, et cetera. And so what we did is just apply our approach to this data for known cell types. Ironically, outside of cell lines, we have very few data sets that have known cell types, and that's a major problem, I think, in this field. We could talk about that. So we did this first for pure B cells. You don't see anything interesting there. Obviously, they're one group, so they should cluster together. But then we did this for the raw data. We were just naive. We're like, Waddington's landscape, it should work. So we put all these cells together, and then we saw this. So what I'm showing you here in gray is the size of the giant component, and you see no steps. And because we know the identity of all of these cells orthogonally, we can, inside that giant component, calculate the number of B cells here in cyan. Purple is the number of NK cells, and blue is the number of monocytes. And you can see here that they all join this giant component more or less simultaneously. We did this with T cells, and we didn't see any differences there either. And we tried a lot of different permutations. We see no separation in the actual raw data for this experiment. And this is just a picture of an epsilon network at some epsilon. The cells are colored the way that I showed you there, B cells in blue, monocytes here, and NK cells in brown. It's a slightly different color scheme, but it doesn't matter. And the only thing I'm highlighting here is that what you notice is that this giant component has all of the three cell types in it. And what this means is that if we look at these B cells in blue, there are B cells that are closer in gene expression profile to monocytes and to NK cells than they are to other B cells. And that's kind of surprising. That would be like finding a cat that was more genetically related to an elephant than it was to other cats, right? That's pretty surprising. So these cells are all on top of each other. And we did this with a number of other data sets. We've now done this with, I don't know, 54 different data sets, and none of the conclusions that I'm going to show you today are different for any of those data sets, basically. We did it for sort of tissues of increasing complexity, the bladder of the mouse, kidney of the mouse, the brain of the mouse, the C. elegans and Hydra. These are C. elegans embryos, Hydra, that organism I told you about before, plant roots. And we see the same thing everywhere we look. So this is kind of interesting. One of the most shocking things for me, because the postdoc lab I was in was the C. elegans lab, and C. elegans has really well-established cell lineages, and it's a eutelic organism, meaning every time development happens, it happens in exactly the same way. And these are the annotated cell types that these researchers sort of determined for each of the cells in their data set. And you can see that this giant component contains basically every single cell type that they identified. So we see no separation even here. There's nearly complete overlap in gene expression space. And so one of the things, so what typically, if you haven't seen this talk before, what usually happens at this point is people start to think about, okay, what are ways where I can explain this finding that Eric is showing to me, but not lose faith in Waddington's landscape. And one of the immediate things that people will often say is, well, look, 10x is noisy, Eric. Everybody says that. What does that mean? Well, lots of different ways you can interpret it, but it's noisy. So isn't it the case that, well, in reality, you have Waddington-like structure in the data, but the noise inherent in the experimental protocol is completely destroying that structure? And that's a question that we can't fully answer because there's no technology that tells us exactly what RNA is in a cell because we don't know, right? But what we can do is look at a bunch of different technologies. So this is a technology called BD Rhapsody that looks at a smaller number of genes, but with a much higher capture rate, meaning it captures more of the RNA in the cell, and we think that should reduce some of the noise. You don't see anything there. This is data that Roy generated on traumatic brain injuries in the mouse brain using a microscopic technique called MERFISH, which is capturing RNA and getting this data very, very differently. This is the same thing, but just with the marker genes that they used in that study. You don't see anything there. This is a different data set that Zach from Roy's lab helped us obtain from the VizGen company. This is the Parse biotechnology with the mouse liver. This is the version 3 of the 10x instrument. So this is a more modern data set where they've improved the chemistry, and you now capture a lot more of the RNA than back when people were doing this in the early days of single cell, and you don't see any differences. You don't see this with protein, you don't see this with the TAC-seq, and there I already told you about the MERFISH. So there is actually a lot of work that we've done to sort of say, well, whatever, I mean, maybe it's noise. Maybe the noise is actually causing this, but whatever that noise is, it's present in every technology we have available to us. So it produces the same result regardless of how you make the measurement, which at least gives us a little confidence that it's not completely artifactual to one type of measurement. Okay, so another thing that people will often ask about is subsets of genes. So one thing that Waddington was never clear, well, actually, Waddington isn't really the person who developed this theory, actually. But Waddington's landscape never said all the genes, right? It's only the important ones, things in the gene regulatory network, et cetera, et cetera. So the question is, can we find subsets of genes that might separate cells? And there are a variety of ways that people do this, even in sort of traditional RNA sequencing analysis. So part of the issue is that you might expect that things like ribosomal genes aren't going to differ much between different cells of different types, but there might be transcription factors or other important markers that will vary a lot. So there are a variety of different techniques that people have developed to kind of, in an unsupervised way, isolate genes that are going to be useful for cell type identification. There's a technique called highly variable genes. It actually typically identifies very lowly expressed genes, interestingly. But we also developed a technique in my lab, and one of the that's called differentially distributed genes. Don't worry about it. And then there's another thing that we did here. So in any case where you have supervised labels, meaning that you know the identity of the cells before you do the experiment, we can actually go in there and do a statistical test to find the gene expression profiles of the genes that are statistically different in terms of their mean expression or something like that between the populations, because we know that these are B cells and these are monocytes. So we can sort of in a statistical way find the genes that are really different. So we're going to call that the Wilcoxon genes because we use the Wilcoxon rank sum test to do that here. But it doesn't really matter how you identify them for what I'm going to show you. So if you use the highly variable genes, nothing happens. If you use our approach, nothing happens. Even if in this particular case with these immune cells, we have the FACS labels, so we can find genes that are statistically different between the populations. And while they are different on average, or in terms of their median or something like that, whatever you choose, their variance is so high that you don't see any separation even in that space. So, I'm glossing over the hundreds of panels of data that we've looked at for various types of selection approaches. We've tried this in every single data set. We've tried a variety of different tools that people have generated for doing this feature selection, and it doesn't change anything at all. Okay, so choosing genes even on the basis of prior knowledge doesn't work. So in this case, as I mentioned in this Murfish study that Roy's lab was involved in or did, they had chosen a set of, I believe, like 60 genes here to be markers of different cell types in the brain. And even if you use those, you don't see anything. You can use things like transcription factors. It doesn't change anything. So most approaches, everything I've shown you has been based on the raw data. And for theoretical reasons that I could go into, you should see if Waddington's landscape, if that modern version of Waddington's landscape, that dynamical system picture of attractors is true, we should see separation in the raw data. But then we got interested because, as I mentioned, clustering is hard, right? And if you've ever done it, you've recognized that like the first time you try it, it often doesn't work. What does it mean by work? Well, it doesn't give you clusters that you like. You know, it doesn't give you clusters that make you happy. So what we thought is, well, let's actually look at how people really treat this data and see if there's separation in any of the kind of spaces that they study. So the way that these pipelines works, as I sort of talked about before, is you have all your genes, you have your raw count matrix, you select your HVGs or you use a different feature selection method. And then what you do is you do CPM log transformation on that. This is normalization and then log transforming the data. I can tell you what that does. It does really crazy things to your count distributions, but whatever, you do it. Then you do PCA, and this is where you do clustering. And people choose the number of principal components using a variety of techniques, but the most common one is to just look at the explained variance as a function of principal component number, and then sort of choose a point that looks like, you know, beyond which you're no longer explaining much of the variance. So it's called the scree plot. And that's what we're going to use to determine the number of PCs to use. You can then compute the distance and do the analysis. So this is no longer, everything I'm about to show you is no longer in the raw space. We're now looking for these discrete jumps in the transform space. And it seemed like when we first did this, that this actually was kind of exciting. So if you do this in your highly variable genes, as I said, it doesn't work. If you just do PCA on that raw data, it doesn't change anything. But then if you do CPM log, you start to see that these things sort of separate a little bit. And then if you do PCA on the CPM log data in this sort of labeled data set that we had from 10x, you do see the steps. So sometimes people get a little confused. Oh, well, maybe you can't see steps at all. Maybe your idea was wrong. But you know, it's actually true that you will see steps if things are separated. We actually proved a little theorem about that. But well, it's really a lemma or the lowest grade of anything you could ever prove, like a nothing, but whatever. The point is that you can see the steps. And there it's like, whoa, hold on, bro, it worked. It worked. Hooray for CPM, PCA log thingy. But it turns out that it doesn't work in most data sets. So most of the data sets that you apply this to, you don't see steps. And interestingly, when they did this experiment, they actually ran all of these purified cell groups in on different wells of the instrument. And so these are likely batch effects, actually, which is interesting. And a lot of clustering tools are validated on this data set still, because it's one of the few data sets of any real biological system where we have orthogonal labels, where you can check to see how your clustering is doing. And it turns out to be really easy to cluster that data. But this data is a lot harder. And one thing I'm just highlighting here is that clustering is hard. So this is that mouse bladder data set I've been talking about. Sorry for the resolution. This is just taken from the study in question. And this is a heat map, and all of these different columns here are different clusters. These are marker genes. And you can see for the vast majority of clusters that these researchers identified, especially the big ones, there's actually no pattern here in terms of expression of these marker genes. This picture actually kind of represents how the data really looks. This is the same type of heat map for the C. elegans data I've been talking about. And there are a few clusters that seem to have very sort of distinct marker gene expression patterns. They're usually the small ones, but these very large clusters, often it's not very convincing. This is just to say that when people do clustering on these data, oftentimes the results that you get are a little wonky and they're not very, very reliable. And Tim has done a lot of work now to kind of characterize how clustering behaves under small perturbations like changing your cell composition and stuff like that. Okay, so that's basically what we found. And we've got, I think now in our supplement for this paper, there's 1,000 panels. So I'm happy to show you. If you have any other ideas, please come talk to me. But one thing that we noticed when we were looking at this data is another kind of surprising finding. So if you look at this mouse kidney data here, you can see that this transition actually is really slow. So I've been working on graph theory for a really, really long time. And in most systems, this transition is usually sharp. Even in random graphs, it's very, very sharp. So we saw this slow sort of increase in the size of the giant component, and we were like, huh, I wonder why that's happening. So we decided to kind of characterize what's going on in this data set in terms of how these cells are actually distributed in gene expression space. And one thing that this picture sort of shows you when you look at this picture is all the cells are at the bottom of the wells, right? And that's what we would expect. The whole point of the height of this landscape is that things should collect at the bottom. Forget about dynamical systems theory. Just as a visual metaphor, if you pour water into this landscape, the water will be at the bottom unless you fill the whole thing. The water will be at the bottom and the hills will be dry, right? That's the basic idea. So we should see that there's some sort of underlying dynamics that leads cells to go to the bottom of these wells. And in a stable attractor, there should be lots of cells near the bottom. Maybe there's a little bit of noise or some fluctuations or stuff, so they're not all identical, but there should be lots of cells here near the bottom and very, very few cells at the tops of the ridges. Otherwise, why draw it this way, right? So what we decided to do was kind of look for whether or not there was that kind of structure in this data. And to do this, we used exactly the same epsilon network approach. What you can do here is instead of looking at these components, you can actually just count the number of cells that are in that epsilon neighborhood. This is essentially looking at the density local to that cell. And here you can see these cells all have 2 neighbors, these cells have 1, et cetera, right? And what we can do then is construct a histogram to look at the distribution of these local densities. In graph theory, this is called the degree distribution. Doesn't really matter. So what I'm going to be showing you is just looking at the density of these regions. All right. And what we expect to see is something that looks a little bit like this, right? So if you have a bunch of cells near the bottom, there'll be a few cells that are in really, really dense regions. Maybe they're closer to the attractor state. They're closer to the absolute bottom. There'll be a few cells that kind of straggle up the sides of the hills. But in general, you should see more or less the same density for all the cells there. So we actually did this in the data. And what we found is this. So, and I'm going to, because I'm running out of time, I'll quickly go over kind of what we found here. So what I'm showing you here is the log of the number of cells that are found at some epsilon in the neighborhood of our cell. So basically, this is no degree plus 1. So at 1, that means that the cells here have no neighbors. These cells have lots of neighbors. Does that make sense? And this is on a log scale, so you can see this goes from having no neighbors up to thousands of neighbors. And what we're plotting here is the probability of finding a cell in the data set that has that many neighbors. And again, this is on a log scale. And what you see is that this forms more or less a straight line, not exactly straight, but pretty straight line on a log-log plot. And so that means that we have lots and lots of cells that have very few neighbors, 0 or 1 or 2. And then we have a few cells that are in extremely dense regions of this landscape. And if we do that for all of the different cell types in these data sets, we see similar things. Interestingly, if you do this on synthetic data, and we can generate synthetic data easily where we sample from attractor-like objects, this is actually synthetic data that was generated by the 10X company themselves, where they just took cDNAs and put it in the instrument. You can see this has a very different shape, and this is what you would expect for kind of a binomial or Gaussian-like thing. This is where you have sort of attractor-like structures for these different cDNAs. But that's not what we see in the real data at all. And it doesn't matter what epsilon you choose. For a wide range of epsilons, you see this kind of scaling result in all the data sets that we looked at. We see it in the mouse brain. We see it in C. elegans. We see it in plants. We see it in every single thing we looked at, all of the technologies. So this is PBMC BD Rhapsody data. You see that same thing. This is MCF10A. This is a cell line. And this, again, this data comes from Roy's group. And you see this in that data set. This is the brain. And you can see here, this is actually the VisGen data from Merfish. You can see this goes from 10,000 to 1. So some cells here at this particular epsilon have 10,000 cells that are neighbors, and others many of them are orphans. And this happens with the BD Rhapsody. It happens in every single modality we've looked at, and I don't have time to go through this right now, but it also happens in every single transformation that we apply. So it's not that CPM normalization or any of that changes this finding. It actually doesn't. This is something that we see sort of universally. Some sort of brief conclusions. The first one is that whenever you're applying dimensionality reduction tools to your data, think carefully about it because it can actually do stuff to your data. And we have one way of characterizing that distortion, but there are actually many out there. You can look at distortion in distances, areas, neighborhood structure, et cetera. There are a bunch of them out there now. One of which we use, but there are many others that have been developed before and since. So you can actually sort of characterize this. And one thing that we found is that no matter what tool you have in your scikit-learn that you're using, they all are really, really bad at dimensionality reduction. All the nonlinear tools, if you give them a sphere, they struggle. And by struggle, I mean not taking the sphere from 3D to 2D, taking the sphere from 3D to 3D. They basically can't do it. And so they're all very, very bad at their job. And I'm not talking about it today, but one thing in my lab that we've done is actually developed a kind of new tool that leverages deep learning and the mathematical definition of this structure, which is mathematically called a manifold. So how many of you have ever heard that term before? How many of you know what it really means? Few of you, but yeah, right? It's like from the Princess Bride. You know, you keep saying that word. I don't think it means what you think it means, right? So people say it all the time, but what does it actually mean? Well, it means that it is possible to cover the set with a set of subsets. Typically, we think of these as neighborhoods, where you can map that local thing to flat space in such a way that you're not distorting the topology. So it's a homeomorphism. So anyway, the point is that we can actually apply that technique. So we built a tool called Deep Atlas. You can check it out on the web or talk to me if you're interested, that actually directly applies that approach. And when we use that tool on single cell data, we see no evidence that it's a manifold at all. So that's kind of interesting. So we could talk more about dimensionality reduction tools if you're interested. But the other and perhaps bigger conclusion is that when we analyze the data, we don't see this expected separation between attractors. And that means that, for one, it seems like the data that we have isn't consistent with this extremely popular picture of how cell types are specified. We can't conclude completely that that picture is wrong, that theory is incorrect, because we need more data, we need more time, we need data on protein levels that is more genome-wide and more effective. There's more work that needs to be done here. But at least as it currently stands, none of the available data that we can find in the literature is consistent with that picture. So that's kind of interesting. And it suggests that either the tools we have are really bad at capturing RNA levels inside cells. So think about it. If Waddington's landscape picture is correct, and your data is as mismatched and as weirdly distributed with this, power law distribution of densities, your experiment is completely messing with the structure of the data. And you might sort of believe that, well, the standard pipeline just undoes all that badness. There's no evidence that it does. So you have to be very careful with that. But the other idea is that perhaps this picture is wrong. And that Waddington's landscape isn't in the gene regulatory network, this space of gene expression profiles. It's actually somewhere else, and we don't know what that would be. Anyway, so one thing that we're interested in is trying to figure out how cell types actually are generated. Nothing that what I said should in any way give you the impression that I think that muscle cells and neurons are the same. They're not, obviously. They're very, very different. But what we don't know now, or at least based on our analysis, is if the data really looks like the structure that we've discovered, how are these things actually specified? It sort of suggests that there may be this paradox that we thought was resolved isn't actually as resolved as we thought it was. Yeah. And one of the other ongoing projects that we're working on, as I mentioned, is how to sort of improve the tools that people use. So Tim is working on how to improve clustering based on these observations. We've worked on dimensionality reduction. We've worked on a lot of different stuff. So if you're at all interested in any of that stuff, I didn't talk about it today because it's still ongoing work, but feel free to contact us. And with that, I'll just quickly thank Brianne and Tim, who did the vast majority of the work. A former bioinformatics graduate student, Seamus Cooley, was involved in that average card distance work that I mentioned. And we started this work with a colleague, Christian Ray, who's now in industry. And with that, I'll take any questions.